about cholesterol in this last section of chapter 13. And cholesterol is a super, super critical molecule in forming cell membranes, in forming some other cell signaling molecules like our hormones. And we know that it's likely to be a contributing factor to cardiovascular disease. So we make cholesterol in all of our cells, but it occurs more in the liver than anything else. It's made in very, very high quantities in the liver. And once we make it in the liver, we send it out through the cardiovascular system, like circulatory. And then it's inside of lipoproteins when it's in the circulatory system. And as long as those lipoproteins are delivered safely to the cells and incorporated into the membranes the way they're supposed to, or maybe used as a substrate to make bile acids or steroids, it's all good. But when our levels of cholesterol get really, really high, then the lipoproteins that are going to be carrying around, so if you have high, very, very high cholesterol, then you're going to have a high concentration of the low density lipoproteins, so the LDLs. And the LDLs are correlated with increases in plaques in the lining of your blood vessels. And those plaques, when they build up enough, then we characterize that as atherosclerosis. And then atherosclerosis leads to blood clots. And if you have a blood clot, you either have a heart attack or you have a stroke. And so making sure that you have the correct amount of cholesterol is really, really, really important. So in this section, we're going to talk about biosynthesis. We'll talk about pharmaceutical drugs that are there to help us regulate how much cholesterol we make. And then we'll talk about the normal regulation of cholesterol in the body. And so we usually make about 1 gram of cholesterol per day. And when we eat, we usually absorb about 0.3 grams of cholesterol per day. So the 1 gram is what we make, and then the 0.3 is what we absorb. And so because we make it, cholesterol is not an essential lipid. So it's one thing you have to really keep in mind. And so when cholesterol is made, it's made in 4 stages. So initially, you're going to start out with 2 carbon units. You're going to start out with acetyl-CoA. So you're going to start with 3 2-carbon acetyl-CoAs, and that's going to turn into 1 C6 molecule. And this occurs with a really, really important enzyme. Let me move this. So with a really important enzyme, and that enzyme is HMG-CoA reductase. Okay. Then, so this is stepwise. It's 1, 2, 3, 4 steps. Okay, so that's our melavonate. That's the C6 molecule. Then we're going to form another intermediate. And this is a really, really important intermediate when you're talking about making plant chlorophylls, when you're talking about making vitamins, when you're talking about making quinones, and that's the isopentenyl diphosphate. And so in this one, you have melavonate that actually gets phosphorylated and decarboxylated. So you have melavonate, it gets phosphorylated, and then it also gets decarboxylated. And if it gets decarboxylated, that means we have 1 less carbon. So we're going to end up with a C5 molecule after step 2. Then step 3 is the formation of squalene, where we have 3 of those C5 molecules that are combined together to make 1 C15 molecule. And then 2 of the C15s, so if you have 2 C15s, they're going to come together and make the C30. And so this squalene is actually also used as a membrane lipid anchor. And we saw that in some different cell signaling pathways that we saw last semester. And then the last step, step 4, you have cyclicization of squalene, where we end up with a 4-ringed molecule. And we're going to remove 3 methyl groups. And you're going to end up with squalene, and squalene is C27. So that's, you can kind of keep track of the carbons that way. All right, so cholesterol. Cholesterol, super, super important. You should know the base structure. You should know that we can take that cholesterol and we can turn it into some different things. We could turn it into bile acids. We can turn it into steroids, right? We could do all kinds of different things with our cholesterol. So if we look at our overview, right, we're going to take 3 acetyl-CoAs, and we're going to take them through stage 1, which is HMG-CoA reductase dependent, and we're going to turn that into melavonate. So this is our 3 C2s. Wow, that's really terrible. Let's do, yeah. Okay, so we got 3 C2s, and our 3 C2s are going to go to a C6. Okay, so now we have melavonate. Melavonate is going to go through stage 2. Melavonate is going to be phosphorylated. So see, we're going to consume our ATP. And it's also going to be decarboxylated. So we're going to lose a carbon. And we're going to end up with our intermediate. Oh, excuse me, y'all. With our intermediate that can be shunted off to go and make vitamins or do whatever needs to be done, right? Okay, so we decarboxylated, and so we're now at a C5, right? The isopentenyl pyrophosphate. Now, we're going to add another 2 isopentenyl pyrophosphates. And there's another way that you could do it, but that's fine. And so we're going to end up with that C15. So we're going to have our 3, right here, we have 3 C5s. Our 3 C5s are going to turn into a C15. All right. And then we're going to turn from our C15, we're going to take 2 of the farnesyl pyrophosphates. So we have 2 of them here. So we're going to get 2 C15s. We're going to turn those 2 C15s into 1 C30. And so our 1 C30 is squalene. And there's lots and lots of steps in here, and you don't have to know all the steps. You just have to know the ones we're covering. All right, so now we have a C30 molecule. And what you can see is at this stage, we don't have the normal steroid structure. There's no steroid nucleus there. And so you're going to go through many, many steps where you go through cyclicization, where you remove 3 methyl groups. And so that's how we end up at cholesterol, which is C27. So our squalene is C30, and then the process form is the cholesterol, and that's the C27. Okay, so how do we make cholesterol? So when you start, you're going to start with 2 units of acetyl-CoA. So they go through a condensation reaction where the CoA is released from one of the 2 acetyl-CoA units. And so you end up with acetyl-CoA, which we've seen before, right? Then we take another acetyl-CoA and we add it with HMG-CoA synthase. So this is the one that's going to be really, really important, because this is our rate limiting step of our entire pathway. Super, super important. All right, so then we end up with HMG-CoA. Then we go through another reductase and we make melavonate. So when we're doing this process, we do need reducing power because we are consuming NADPH. And we're also going to release our coenzymes, right? So every time we're going to release 1, 2, 3 coenzyme A's. So if you want to look at, if I'm a pharmaceutical company and I want to regulate how much cholesterol a person can make, well, I want to look at my rate limiting step of the pathway. Well, that's HMG-CoA synthetase. And so if we target this one, this is called a statin drug. So the HMG-CoA synthase is really, really important. Did I say? Oh, y'all, I circled the wrong one. This is our rate limiting. Here we go. This is our rate limiting step. Sorry, y'all. So HMG-CoA reductase is our rate limiting step. So this is the one that we target with our statin drugs. So the whole point of limiting the activity of HMG-CoA reductase is you will not make melavonate, and so it will not go on and make more cholesterol. And so it helps to reduce our risk of cardiovascular disease because we decrease our flux through the cholesterol biosynthesis pathway, right? You could only then get cholesterol from your diet. You wouldn't be able to make much of it at all in your body, right? Okay, so that's stage one. And the end of stage one is where we have our rate limiting step. So stage two is where we're going to start out with melavonate. Our melavonate is going to be phosphorylated, right? So it's going to come from ATP. And then we're going to phosphorylate again at the same site so that we end up with a pyrophosphate. Remember when we talk about pyrophosphate all by itself, we say it's PPI. Well, it's pyrophosphate now attached to melavonate. So once we add our phosphate groups on, then we're going to go through a decarboxylase reaction where we're going to lose our hydroxyl, and we're going to lose the carbon right here. And so then we'll end up with isopentenyl pyrophosphate. And the isopentenyl pyrophosphate can be used directly, or it can turn into another substrate. And they can kind of convert back and forth unassisted by enzymes. But these are basically what we call these are isoprene units. So here's either one of those have isoprene units, but isoprene, this general structure is a common building block for lots of different things, right? And just remember that this is ATP dependent, right? So you're going to have to go through quite a bit of ATP in order to make your cholesterol, right? All right, so then in stage three, we have our isoprene units that are attached together, and then you have two C5s come together to make one C10. Then we attach one more C5 and we make our C15, which is farnesyl diphosphate. Then we're going to take two C15s and we're going to form squalene. So squalene is our C30, right, molecule. So if we look at our steps here, right, we're looking at stage 3A. So the first thing is that we're going to do a condensation reaction in what's called a head to tail orientation. So we're looking at the phosphates as being the head groups. And so what we're going to see is we'll have a head attaching to the tail portion of the other molecule. And that's why they call it just a simple head to tail condensation. So then now that we have a molecule that has 2 units attached to it, now we're going to add yet another C5. So we have another isopentenyl pyrophosphate. Ooh, trying to erase that. Okay. So now we can make our farnesyl because we now have 3 units, the black one, the red one, and the blue one, right? And so now we have our C15 molecule. Now we can go to stage 3B where we go through squalene synthetase, and squalene synthetase is going to add 2 C15s. So we have one C15 here, and we have another C15 here. And so those are going to get joined together. And in this case, you're going to do a head-to-head addition because you see the blue head and the blue head. You're going to do this one in a head-to-head fashion. And so you got to think about, gosh, this is consuming so much energy. What is going to drive these reactions forward? Well, every time that you do this, you're releasing pyrophosphate. See how we release PPI? If I can erase all these circles, releasing PPI. So remember that the delta G value for going between PPI to 2PI is a very negative delta G. And so the hydrolysis of pyrophosphate is what helps to drive those reactions because they have a very, very negative delta G value. So once we make our squalene, then we can go in and cyclicize those molecules, and we'll get our normal steroid nucleus that we're looking at. And it happens with a lot of cyclases. It's a lot of steps that you don't have to know. It's like 19 steps in here. So it's a very complex set, and we don't really worry about that so much. You just have to know what has to be done. You just have to cyclicize it, and then you have to remove some of the methyl groups, and you're going to get your final product. So no big deal. All right, so if we have cholesterol and we esterify our cholesterol with a fatty acid. So if we have cholesterol, we esterify it with a fatty acid, I mean, you'd have to have some kind of like acyl-CoA cholesterol acyl transferase. You're going to make a cholesterol, yeah, Joe, a cholesterol ester, right? And so once you have that cholesterol ester, now you can do different things with the cholesterol. You're typically either going to immediately embed it in a membrane, or you're going to turn it into an ester. And so there are three major functions if we turn it into an ester. Number one is just storage. We're going to store it in those lipid droplets where we stored the tricarboxylic acids, right? We could package them up in lipoproteins, export them out to whatever cell type needs it, no big deal. And then you can also secrete it into the small intestines through the bile duct. So this secretion right here, this is really the only way to rid the body of excess cholesterol. So rid excess. So bile acid can be recycled back to the liver, but some of it is going to be excreted as waste. So one way that the pharmaceutical company has done this is you supplement your diet, it's kind of cool, with an indigestible resin. And that indigestible resin binds to bile acids with super, super high affinity. And because excreted bile is not recycled, then the liver sends more cholesterol into bile acid synthesis. And so it basically decreases the total cholesterol ester pool. But you have to consume quite a bit of it. You have to consume like, I don't know, it's like 10 to 20 grams of resin per day. I don't know if I'd be on board for wanting to do that, but it works. It actually works. So it's kind of cool. All right. So what are we going to do with our cholesterol that's from the liver? And remember, they're esterified. Woo. So we have our cholesterol esters. And both triacylglycerols and cholesterol are transported through the circulatory system as components of our plasma lipoprotein particles. So I don't care that you know all of these different steps. You just have to know that you're eventually going to turn into bile acids. You don't have to remember any of those steps right there. Okay, so we talked about this in the very beginning, that if you had excess cholesterol in those very low density lipoproteins, leads to plaque buildup. The more plaques that build up, the thicker it gets, then we can kind of classify it now as atherosclerosis. And so when you have atherosclerotic plaques, basically red blood cells kind of get trapped on them. And then you end up piling more red blood cells, and it turns into a clot. And then the clot blocks the entire blood vessel. And so now no red blood cells can get through. And so then you have increased cardiovascular risk, right? So when you look at this, you got to think about what's transporting our cholesterol everywhere. And we know that we need lipoproteins inside of our vesicles that are carrying all of these particles around the entire body. And so we call them apolipoproteins. So these are the ones that are membrane bound in the vesicles. And then they have that core that faces inwards to those cholesterol esters or those tricarboxylic acids, right? And so the phospholipid monolayer can have the free cholesterol, but the esterified cholesterol is in the middle. And a lot of times it actually serves as a signaling molecule, right? And depending on the ratio of protein to triglycerides will tell you kind of like, oh, these are higher density, these are lower density. And so they have different functions, right? So the functions of our lipoproteins vary, and they depend on the type of apolipoprotein and the lipid cargo that they carry, because that changes their density. And so they behave differently. So we've talked about all these different kinds before, but now we're going to kind of give them functions. So if you look at the choliomicron, our choliomicron is going to be our largest in diameter. But when you look at its density, it's actually the least dense. And it has the lowest percentage of proteins. Proteins tend to be very, very heavy. So then if you look at the triacyl glycerol molecules, oh, that's a lot of triacyl glycerol molecules, right? So these are mostly, so our choliomicrons, if you kind of write underneath, these are mostly triacyl glycerols, right? That's because they're low density. If you have more fats and less proteins, so you have low protein, that's going to give you a very low density. So choliomicrons are that first class. Then we have very low density lipoproteins. So these are our VLDLs, right? Our VLDLs are smaller in size, much smaller in size, but they have higher densities. And if you look, well, what happened? I went from 2 to 10. So my percent of proteins went up. My percent of triglycerides went down, and my percent of cholesterol esters went up. So we can see that they have different cargo, and so they have different properties. Then you have intermediate. So these are IDLs, intermediate density lipoproteins. And so they're a little bit more dense, right? A little bit more protein, a little bit less triglycerides, and a little bit more cholesterol esters. So as we're kind of going across, if you look at your cholesterol esters, what's happening here, we keep going up, going up. Now, high density lipoproteins are different. We're going to talk about that. Then the next one is low density lipoproteins. So these are LDLs, right? And then you have HDLs, high density lipoprotein, HDLs. So remember LDLs, HDLs, right? So if you look at HDLs, wow, look at the percent of protein there. 55%. That's a lot. But you have very, very low lipid concentrations. It's the highest density, it's the lowest amount of lipid. And this is really kind of interesting because the HDLs are really, really good for you because they return cholesterol to the liver. So if there is excess, it's bringing it back. Well, I want more of that, right? I want more of the scavenging molecules so that they'll go in and get the excess cholesterol. And so I don't make plaques. So that's why when we talk about HDL and LDL, that's why it's important. So this is just a reminder of what they look like. But remember, it's not about size, it's really about density that's going to tell us how it functions. Choliomicrons are huge, but they're very low density. Then if you look at HDLs, oh, they're very high density because they're made majority of proteins, right? And that kind of stuff. The only one that we haven't talked about yet are the choliomicron remnants, and these are just formed as choliomicrons drop off their cargo. So they're not really transporting proteins. They're just like the after effect of dropping off the cargo. All right, so let's talk about what happens when we need to move around our lipoproteins. So if you think about the steady state levels of circulating cholesterol, they have to be balanced by a couple of different things. We're going to balance it by input. And input can come from one of two things. It can come from your diet, you eat it, and it also comes from synthesis, right? It has to be dependent upon our cholesterol recycling, right? So cholesterol recycling. So our recycling is how much is left in the tissues that comes back to the liver. So that's like our HDL particles that are doing that. Then we have our cholesterol output. So where are we losing cholesterol? Well, the only place where you're losing it is from bile acids. That's the only place. So when we look at what happens, you're going to see all these pictures, and what's kind of interesting is that each one has a different size. Well, if you look at the size of the lipoproteins, the size of the lipoprotein is decreasing as our triacylglycerides and our cholesterol cargo are transferred from a lipoprotein particle to endothelial cells that line our circulatory system. So our liver here is doing the de novo cholesterol synthesis and recycled synthesis. So here's our liver, and we're going to make some things. All right. Once we've made it, the output is either that we're going to excrete it as bile acid. So that's kind of one thing that could happen. Or we're going to send it out in like choliomicrons, right? So they're going to go into the circulatory system. The fatty acids are going to be released. We're going to utilize them, whatever it is, right? So either way, we're going into the circulatory system. Once they drop off, then you get remnants of those really big choliomicrons. You get intermediate density lipoproteins. As they continue to drop things off, you'll get low density. The only one that's kind of weird that goes in reverse is our HDL precursors. So these HDL precursors go out into the bloodstream. They don't have anything in them. They're mostly just proteins. And they pick up tissue cholesterol, and then they bring it back to the liver. So that's why we call it the good cholesterol, right? So our good cholesterol, our HDL particles, these are those reverse ones, right? And so if you look at this, you can look at the different apolipoproteins that are apolipoproteins that are embedded in the membrane, and you can see exactly which class they're in. But if you just look at their mass, you know. Right? And so these are the different kinds of lipoproteins, but I don't expect you to know that. What is important is if you want to know, okay, do I have to worry about my cholesterol levels, which you're not actually testing cholesterol levels, you're actually testing HDL or LDL levels. So we know if you go on a 12-hour fast and then you check somebody's cholesterol, if you check their cholesterol after that 12-hour fast and they have high, low density lipoproteins, they are at a high risk of cardiovascular disease. But if they have high HDL, which we know is the recycling one, then they have a low risk of cardiovascular issues. And so that's why LDL gets the name the bad cholesterol, whereas the HDL gets the name the good cholesterol. And it's not completely and totally understood why, but we do know it's related to function, right? And we know we have to worry about this. Like we really have to worry about this because cardiovascular disease is the cause of half of all deaths in the US, right? And the main problem is that you have blockage in the arterial system of the heart. And so remember, the arterial system of the heart is what's feeding the heart blood, oxygenated blood, and it gets blockages because of those atherosclerotic plaques. And so then you end up getting the fatal heart attacks, you get strokes. And if you look at those plaques inside of the people's blood vessels, they have these massive deposits of cholesterol-rich lipids, right? So what we know is there's some kind of infiltration of the low density lipoproteins into the space like below the endothelial cells. I don't think they had a picture of it in your book. Yeah, they don't really have one in there, but it gets below the endothelial lining of the blood vessel. And so then the blood vessel kind of starts to break down. So it's really not good. And so they think basically what happens is the phospholipids, I find this stuff really super interesting, get oxidized in the LDL particle, right? And it's part of the innate immune response why that happens. And so macrophages, they call them foam cells, or a type of macrophage, go in and try to degrade the low density lipoproteins, right? But when you try to degrade a low density lipoprotein and it opens up, it releases its cholesterol. And then that cholesterol gets deposited in the plaque. And then you have all of these things are saying, you know, something's hurt, something's broken. And so you have this body trying to repair itself. And that's happens with smooth muscle cell proliferation. So those cells that line the blood vessels try to grow and basically heal, but it doesn't work, and it gets calcification, and then the plaques can rupture. And if you have a rupture of a plaque, then you have this fibrous blood clot that forms and blocks the blood flow. And so you got to think, why are we always so worried down here about high blood pressure? Well, high blood pressure increases our risk of arterial damage and plaque formation, right? The higher the pressure of the blood is, the more you risk damaging the arterial system and opening up those plaques. So if you eat a diet high in saturated animal fats, well, the more you consume, you're going to lead to elevated serum low density lipoproteins, LDLs, and you're going to get plaques. So you can do a simple blood test to see what your LDL levels or HDL levels are after you do a 12-hour fast. That's kind of the way to do it. But you should see there's not really. There are interesting cases where people have had mutations in the LDL receptor gene. So they have basically no receptor sitting on the outside of the cells. So the cells that are normally supposed to take in LDL don't do it. And so their serum LDL levels are like through the roof. They're like 5 times what the normal should be. And so these are people who have heart attacks like as children. It's ridiculous. It's terrible. But it's also interesting. Okay, so I still go on a side track even when y'all aren't in class. So there are 3 big processes that control our cholesterol homeostasis in our liver cells, and your book has kind of classified them. If you look at the numbers, you'll see the numbers are in different colors. So the first one, the black ones are endocytosis. So this is endo, I don't think they say this, maybe. Endocytosis of our low density lipoproteins by LDL receptors. And this is what I was telling you about, that the patients just don't have these little receptors right here, these little green things. They just don't have those. And so then their blood serum levels of those LDLs are crazy. The blue ones, the blue numbers here, see if I can come out here. The blue numbers are de novo, and de novo just means new, like you're making it new. Cholesterol biosynthesis. And where are we? You have to always remember where we are, and we are in the liver for this. Liver. Okay. And these blue steps, this is what we typically target with statin drugs. So this is targeted with statins. Okay. And then the green ones, and I'm going to go over each one. This is the stimulation of the LDL receptor gene expression. So this is how much receptor we're putting out there. So this is stimulation of LDL receptor gene expression. And so what triggers this? So if you look at why would you want to make the cells take in low density lipoproteins, take in things that have cholesterol? Well, that's only going to happen if you have low intracellular cholesterol levels. So this is intracellular, right? Because the cell needs some cholesterol in order to function. It does. And so when you have low cholesterol, what you end up doing is you end up activating this transcription factor, SREBP. So this is the transcription factor that controls expression of the LDL receptor gene. So once it becomes activated due to low intracellular cholesterol levels, we increase transcription of the gene, then we make the mRNA, ship it out, make the receptors, and then ship the receptors out to the outside of the cell. And so this is what ends up promoting, now that you have these you have all these receptors, they're ready to go. Now they can bind LDL. So this intracellular mechanism is what increases LDL into cytosis, right? So now that you have the receptors, they can bind to the LDL particles, they get internalized, right? And then this is just a clathrin-coated pit. It's a way for the cell to make a pit. And the clathrin gets recycled, but the vesicle, it actually breaks off. And so you end up with two pieces where you're going to recycle the receptors up to the surface. And then our contents of the LDL are going to keep going and going to be processed. And so it kind of depends on what you need, what you're going to make. You might use it to make some amino acids. You might use it to make fatty acids. You might use it in those cholesterol esters to break those down and then contribute to the cholesterol pool, so we can go make more bile acids, we can go make more steroid hormones, we can go and do whatever it is that we need to do. And then we know that the majority of the cholesterol that's in our cells, remember we said this, is actually controlled by this blue mechanism. And the blue mechanism is where we go from acetyl-CoA and we make our cholesterol. And that's 1 gram versus 0.3 grams, right, from your diet. So we want to target the part of the system that makes the most, right? All right, so these are examples of the statins. There are a couple of different kinds, but they kind of mimic some kind of part of HMG-CoA, so that they can reduce the serum LDLs by pretty significantly, like 20 to 40%, because you're going to inhibit that HMG-CoA reductase. But they also have an effect of stimulating LDL receptor expression, right? So if your intracellular cholesterol levels go down, then you're going to promote LDL receptor expression. And so you increase those LDL receptors, but they don't work in all the patients. And when you end up having to increase somebody's dose over and over and over again, you get unfortunate side effects. One of them is muscular degeneration, and there's some other side effects. So you have to be very careful with statins, very, very careful. So remember how we talked about there's a transcription factor that responds to the levels of cholesterol intracellularly. And so there's a set of enzymes that are required to regulate this cholesterol flux, right? And so it makes sense. All of our lipid metabolizing enzymes should be under some kind of umbrella transcriptional control. And so there's one transcription factor that controls a whole set of functionally related genes. And we call this the steroid regulatory element binding proteins, or the SREBPs. So this is sterol regulatory element binding proteins, right? So these are all proteins that are embedded in our ER. And so if you have a lack of cholesterol inside the cell, then these transcription factors, these SREBPs, are going to bind to sequences, the same sequences in the DNA, and those sequences are upstream of the LDL receptor gene, the HMG-CoA synthase gene, and the HMG-CoA reductase gene. So all of these different gene products are under the transcriptional control of one set of transcription factors. And so all of these enzymes are, of course, in the mechanism for cholesterol biosynthesis. So they're all going to, activity of all three of these genes is going to end up increasing our levels of intracellular cholesterol. And that's what we want to do, right? Okay, so if you look at SRBP binding, how does this work? It's really kind of interesting. So all the pieces are there, but they're just not available to work yet. And so this is our DNA portion of the transcription factor that is going to go in and bind to the DNA and promote expression of the genes. But it's actually in its precursor form where it's embedded in the ER lumen. If it's embedded in the ER lumen, trust me, it's not going to the nucleus. And so this is what our cell looks like if we have lots of cholesterol, high cholesterol. If we have high cholesterol, we have this protein called SCAP and NSIG that basically keep things shut down. So if you have enough cholesterol, both of these things are bound to cholesterol, and they basically trap our transcription factor in the ER membrane. So when cholesterol levels start dropping, there's no more cholesterol bound to our NSIG or to our SCAP. And so basically they start disassociating, the pieces start disassociating. And then after the disassociation, you have to notice here, we're in the ER, but after the disassociation of NSIG, then our precursor transcription factor and the SCAP protein get translocated to the Golgi. Once it's in the Golgi, there's an enzyme there in the Golgi, S1P, that can cleave our precursor protein. And so once that cleavage event happens, now our transcription factor is no longer bound to like the silencing complex. And so now a second enzyme can go in and cleave off the transcription factor head. So you need two cleavage events. And once that cleavage event happens, now you can have that transcription factor go into the nucleus and go and bind to the correct gene sequences and then start the whole system. So I want to kind of come back here. That's like the last slide, but I want to kind of talk about how statins really work, right? How do they lower cardiovascular disease? So remember we said that they come in and they're going to block, right? They're going to prevent de novo synthesis of cholesterol via our HMG-CoA reductase. And so if we have lower levels of intracellular cholesterol in our liver cells, right, it's going to trigger our transcription factor to go in. First, the precursors, right, in the ER, goes to the Golgi, gets cleaved. The transcription factor goes to the nucleus, promotes expression of our LDL receptor and all the enzymes involved in cholesterol synthesis. It's not the only thing. But here's the great thing, is that it's going to lower the serum LDL levels, because if you have higher receptor, LDL receptor expression, you're going to have more of those LDLs pulled out of the bloodstream. So if you pull out more LDLs, you're going to have less LDL deposition inside of the blood vessels. You're not going to have the foam cells that are going to try to break down the cells and break open the cholesterol. So we won't have plaques, we won't have ruptures, we won't have blood clots, and we won't have heart attack or stroke. And so they're working on some new alternatives to the statins, because the statins do have a lot of off-target effects. But for now, it's kind of like the go-to in cholesterol maintenance. All right, that is the end of chapter 16.